Thank you very much, Ifeoma, and firstly to thank KDIGO for inviting me to this wonderful conference. I'm really looking forward to the next few days, and I think it was lovely to hear Dr. Friedman give that very elegant talk, telling us about some of the mechanisms, but also raising a lot of questions. As he said, lots of research for the next 10 years or more. So I have no disclosures except to say how happy I am to be in Ghana and to feel very welcome, because this is one of the countries that permits me visa-free travel. So the moment you present yourself at immigration, you feel highly welcome. So thank you, Ghana. I'm going to try and address some of the listed outline for the epidemiology, but in 20 minutes I've had to condense a lot of the evidence, and I'm not able to present a lot of the evidence. The next two slides, basically, I put in to tell us how APOL1 evolved, and the whole story of APOL1. Long before APOL1 was reported, I think clinical researchers in the field noted certain aspects of kidney disease that were associated with African ancestry. For instance, we were aware of focal segmental glomerulosclerosis occurring in people predominantly in people of African ancestry, and also associated with it steroid-resistant nephrotic syndrome, and then so-called hypertensive nephrosclerosis and HIVAN. And there were lots of theories as to why this could have happened. And then, of course, the USRDS database raised some questions. For instance, the fact that chronic kidney disease was fourfold more prevalent in people of African ancestry than in those of European ancestry. And then also the whole question of HIV kidney disease, and especially HIVAN, being 18 to 50-fold more prevalent in people of African descent. And this basically results in the whole outpouring of APOL1 research. And I won't go into the slide very much, except to point out the prevalence of the different genotypes. If you look at the one risk variant, globally it was reported that 37% had this, whereas the high-risk 2 gene variant occurred at about 13% globally. But of course, there's a huge geographical difference. For instance, we're in the heart of APOL1 territory in West Africa. If you look at the highest numbers in Nigeria, in the Yoruba, and in Ghana, among the Asante, over 40%. And then, of course, in the rest of West Africa, there are some preponderance in Cameroon, up to 15 to 16% of these variants. Central Africa, otherwise, much less. And then if you look at the rest of Africa, you can see that it's hardly been reported in North Africa or East Africa. And again, small numbers in Southern Africa, or maybe not so small if you think about 12% being a respectable number. And then in the rest of the world, you can see that APOL1 has hardly been reported, except in countries with populations of African ancestry. And if you look at African Americans, where the highest prevalence of just over 22%. This very elegant slide shows how these genes evolved. West Africa with Trypanosoma gambiense, East Africa rhodiense. And you could see that the APOL1 G1 frequency is highest in West Africa, as I've shown the figures in both Nigeria and Ghana, and a small focus in Southern Africa of between 10 and 20%. And if you see, East Africa hardly has any reported preponderance of APOL1. And this really nice slide talking about human diversity and migration shows how the APOL1 gene was taken to other parts of the world. Firstly, the within Africa migration to Southern Africa, and then the out of Africa migration with the slave trade towards the Americas. You've seen this slide previously, but just to point out that the highest effect of APOL1 is in interferon-associated diseases and in HIVAN and focal segmental glomerulosclerosis. This slide that we published recently, looking at APOL1 genotypes in different African populations compared to the US, you could see that in HIVAN per se, there was a very high odds ratio of 29 and a lower one of 3 in these two populations, whereas in Southern Africa we reported one of the highest associations with the odds ratio of 89. And a recent study that was published by our colleague from KANU, again, these are all biopsy-proven studies, showed that they had a much lower preponderance of HIVAN with their odds ratio of 5.5, whereas they had a higher prevalence of focal segmental glomerulosclerosis with a higher odds ratio of 9. And then this recent study from the Awijan study, in one of their tables, they summarized the various high-risk genotypes associated with kidney disease in the USA and sub-Saharan Africa. And again, you can see that HIVAN has the highest associations. But interestingly, in pediatric populations, especially in the DRC, there seems to be very high prevalence and association with proteinuria, and I'll present some of that data subsequently. So I've summarized some of the conditions that are associated with APOL1. If you look at hypertension, Genovese reported in 2010 that this was 7-fold odds ratio, whereas in the study of African American study of kidney disease, this was 2.57, but interestingly increased to over 6 when advanced CKD ensued. And everyone's aware of the data with focal segmental glomerulosclerosis with a 17-fold risk, and the HIVAN's ratios are well known. Looking at it again in children, one saw that in children with perinatal HIV infection, there was 3-fold increase odds of chronic kidney disease with a high-risk genotype, whereas if and they presented at an earlier age with a high-risk genotype at a median age of 8.8, whereas those with 0 or 1 risk allele, their disease only presented at the age of 14. There's a range for lupus nephropathy from 2.7 to 5.4-fold risk, and sickle cell nephropathy at 3.4. I thought this study that we did was quite interesting because this teases out the risks of the different G1 genotypes with the different populations that we studied. These were all biopsy-proven diseases, HIVAN, the various chronic kidney diseases associated with HIV, and those that were not present in people with HIV. And if you look at the association with the 2-risk genotype, one can see that the heterozygous G1, G2 at the highest percentage at 50%. And overall, just under 79% were homozygous G1, G2, or compound heterozygous, whereas there was a 3.7% prevalence in HIV-positive controls, and our population control had just under 2% APOL1 positivity. Again, in concordance with other studies showing that that APOL1 risk does not really manifest in South African populations of African ancestry. Looking at this interesting Gen Africa study published in the UK, looking on your right, you can see the distribution of APOL1 genotypes, black and gray showing the G1, G2 admixtures, being highest in the West African and Caribbean populations of African ancestry. And then the next section showing the increase of this genotype in people with end-stage kidney disease, where there's a huge association with the G1 genotype. Looking at it in children in the DRC, one could see that in children with HIV, with the high-risk genotype, there was very high prevalence of proteinuria of 80%, whereas in the general population and in those without the high-risk genotype, proteinuria was minimal. And looking at it in people with HIV in West Africa and Cameroon, this paper reported no direct association with APOL1 high-risk genotype and with GFR change over time. But the authors did report that those with the high-risk genotype and high basal viral load had a faster eGFR progression. This is an interesting study from the Awigen cohort that looked at over 10,000 people from 4 countries, from Ghana, Burkina Faso, Kenya, and South Africa. And one could see that the G1 alleles were in the highest frequency in West Africa, as expected, but surprisingly also in Southern Africa in the study, whereas the G2 seemed to be commoner in South Africa. And this was a young population with a mean or median age of under 30. And they found that there was association with albuminuria in the study. And interestingly, the albuminuria association was strongest with the G1 genotype, with the odds ratio of 3.87 compared to about half or less than half that with the G2 phenotype. Now looking at outcomes with APOL1 status, this old slide shows this quite elegantly, where one can see that those with 2 risk alleles progress much more than those with 0 or 1 risk allele. And this was also shown in the African American study of kidney disease, where those with the 2 copies of the APOL1 risk variant also progressed more rapidly over time, also irrespective of proteinuria status, and also was related to blood pressure control or antihypertensive drugs. And looking at this recent study, looking at the associations and kidney outcomes, one could see that in both univariable and multivariable analyses, there was greater progression to end-stage kidney disease in those with the APOL1 high-risk genotype. And similarly, the association with proteinuria and albuminuria was reported, as well as that with chronic kidney disease as defined by an eGFR of less than 60. And also the associations were reported with different histological or clinical diagnoses, such as FSGS and HIVAN. And in fact, the highest association was in HIVAN with the 34 odds on multivariable analysis. In this slide, I just want to point out that as long ago as 10 years ago, it was found that those with high-risk genotype in black populations had a higher prevalence of albuminuria, as well as a faster eGFR decline over the study period. And similarly, in children, COP also reported that the APOL1 high-risk genotype tended to present at an older age, and these patients had a lower baseline eGFR and more segmental glomerulosclerosis and total glomerulosclerosis, more tubular interstitial disease, and more collapsing variants. This recent study in children, looking at children with steroid resistant nephrotic syndrome and FSGS that was biopsy proven, and progression to advanced CKD, one could see that those who had the high-risk genotype had a stronger association with an odds ratio of 2.86. This slide, my thanks to Dr. Friedman and Skorecki for this. And if one looks at populations with APOL1, not everybody progresses to the disease phenotype. So the theory of disease modifiers or second hits was proposed, and the slide shows the various disease modifiers. For instance, viruses, HIV, and more recently we were aware of COVAN, reported in populations with COVID, found to be associated with the APOL1 genotype. And then viruses that may modify the disease, like the polyoma virus. And of course, we're all aware of interferon as being one of the strongest promoters of disease. And also the effect of antiretroviral therapy. And there's some interesting clinical data coming up with antiretroviral therapy. So looking at the response to antiretroviral therapy, we're aware of the fact that before antiretroviral therapy was available, HIV kidney disease, especially HIVAN, was a death sentence where people progressed very rapidly to end-stage kidney disease and death. And since therapy was available, it's become a chronic condition that can be managed. And the next slide by Estrella's group showed how important effective treatment and viral suppression is to preserve kidney function. So if you look at the first graph showing that if there wasn't viral suppression, the disease progressed quite significantly over an 8-year follow-up, whereas if there was viral suppression, patients' kidney function was very well preserved. On this busy slide, I just want you to look at this line. These are all biopsy-proven data from 2 South African centers, Johannesburg and Cape Town, and a US study with quite large numbers of patients. And you could see the classic HIVAN numbers are really markedly decreased, whereas previously prevalences of over 70% were reported in HIV population. And when we really wanted to study HIVAN, we actually struggled to find this disease histologically. And in our study, we found that over this follow-up period that we're currently reporting about a 25% HIVAN in our biopsies. And this is all thought to be treatment of the general population because lots of people still presenting to us are treatment naive. So that is something that's quite interesting, and I can't quite get my mind around it as to why we're not seeing so much of HIVAN in treatment naive patients. The protective P264K variant is very interesting, attached to G0 and G2. And the suggestion made in this recent paper is that one should redefine the risk stratification based on this gene, that the high-risk genotype should be those that do not have this risk variant. And this slide shows very elegantly what happens to people who have the risk, who have this variant or do not have the risk variant. And you could see that there's almost a 2-fold increase of chronic kidney disease in people who do not have the risk variant, and almost a 4-fold increase of end-stage renal disease if these populations do not have this risk variant. And a recent paper looking at a genome-wide association study of chronic kidney disease. And we're all aware that uromodulin is one of the important risks for chronic kidney disease. And this shows that this has a very high risk for CKD in these populations. But as Dr. Friedman says, there's lots more to be known and to be reported on in the next few years. just to thank my colleagues at WITS and the University of KwaZulu-Natal, whom I worked with over many years, and to thank Jeffrey Kopp, Cheryl Winkler, and George Nelson, who set us off on our adventures in APOL1. Thank you very much for your attention.
Question for Dr. Naicker.
Sarala, thank you for a wonderful talk and really reviewing this whole field. I have one question, and that's Trypanosoma brucei rhodesiense is commoner in Western Central Africa, but the variant, APOL1 variants that protect against Trypanosoma are more common in West Africa. And it speaks to me that there must have been migrations, which actually I couldn't pick up in the figure that you showed of migration, because much of the migration has been from west to east. So that's one of the things that's always puzzled me, why the APOL1 risk variants are so much higher here. I'm not saying there's an answer, but do you have any thoughts about that? Thank you.
I looked into it because we did discuss this previously, and I think there's been a lot of within Africa migration from west to east and east to west and to the south. So I'm not sure how that would have affected the APOL1 variants, or whether there's another sort of protective gene, for instance. I don't know. I'll be very interested in input from our geneticists to see if they have any inputs on that question.
You have your mic.
No. There it goes, thank you. Hi, Sarala, thank you very much for that excellent talk. What is the prevalence, do we know the prevalence of APOL1 variants in the San population of South Africa?
It was reported, I think, at around just under 20%, if I recall, but I didn't particularly pick it up on this. And they don't seem to have so much of APOL1 reported kidney disease in the form of, say, in the HIV, for instance, but more sort of hypertensive attributed kidney disease and probably FSGS. I think there's a question.
Is there a difference in prevalence by biological sex?
Sorry, I missed that.
Is there a difference in prevalence by biological sex?
By biological?
Sex.
Steps.
Male, female?
Probably yes. If you look at HIV populations, there seems to be more HIVAN in men, yet women seem to have the infection more. But I'm not sure about the mechanistic reasons for that.
Doctor, have you yet seen any evidence of differences in the distribution of the protective variant? Do we know anything about that?
We haven't studied it as such, and I think that should be something that people need to look at, because that's a very interesting possibility, isn't it?
Right.
Whether women have a higher prevalence of the protective variant. So I think it's a very interesting question. Thank you. I think this one.
Thank you very much, Dr. Naicker. I think
Thank you.